Today I am going to explain dissolved oxygen experiment. Generally, as we breathe air from the atmosphere, in the same manner the aquatic organisms they need the dissolved oxygen in the water. So, for that experiment we need the materials required: burette, burette stand, MnSO4, potassium iodide solution, starch is the indicator, and the sulfuric acid, and then water sample. For this experiment, we have to collect the water sample in a specific water bottle. We can call it as a BOD bottle or DO bottle. So, while you are collecting the water sample, you have to dip the bottle completely inside the water sample, and you have to fill the completely bottle without any air bubbles. So, since we are doing the dissolved oxygen experiment, we should avoid the air contact from the outside to the inside water sample. So, collect water sample in this bottle, and then first we have to add the first reagent MnSO4 2 ml with the help of this micropipette. After adding MnSO4, next you have to add the potassium iodide solution 2 ml. So, once you add these two reagents, the MnSO4 and potassium iodide solution will react with the water sample, and it will appear red color precipitate. This indicates that dissolved oxygen is present in this water sample. So, the excess solution, keep the cap on the bottle and remove the excess solution in the sink, and mix it twice and keep it aside to react with the water sample. If it is white color precipitate is formed, that indicates that there is no dissolved oxygen present in the bottle. After forming the red color precipitate, then we have to add the concentrated sulfuric acid 2 ml to this bottle. So, after adding this concentrated sulfuric acid, the precipitate will dissolve, and then we have to mix it well, and we have to remove the excess solution in the sink. Now, we have to mix it well to dissolve the precipitate. Now, we have to take 100 ml of this solution into the conical flask by using measuring cylinder. Now, we are going to start the titration process against sodium thiosulphate, which is loaded in the burette 0.025 normality. Start the titration process until light yellow color. Stop the titration process once we get the light yellow color, and then we have to add the starch indicator. So, when you add the starch indicator, the solution turns into blue color, and then continue the titration process until colorless. Colorless is the end point in this titration process. Stop the titration process when you get the colorless end point, and note down the burette reading, start point and end point. Now, we have to do the calculation by using these burette readings. So, the normality is 0.025. So, repeat the steps to get the concurrent readings. So, the dissolved oxygen units can be expressed as a ppm.